We're going to talk a little bit about how powerful that plant is for changing that soil ecosystem. We're going to go over a little bit about that, and then what we're going to do is I'm going to work our way towards the Haney test. And we're going to talk about that, the Haney test, and how we can use that. And so, to me, that was one of the most powerful things that really helped me. It was one evening, I keep telling the story about, it was one evening I was reading Dr. Michael Swift, a very great researcher. And I was in the back of my bedroom and I was reading this book. It's called Decomposition in Terrestrial Ecosystems. Excellent book. It's about a $200 or $300 book, very hard to come by. And he was saying, is the plant and soil are one? And I got really excited. It was kind of like an epiphany moment for me, Neil. And my wife says to me, I'm screaming in the back, and says, well, she goes, what's wrong with you? She goes, hon, the soil and plant are one. She goes, oh, I thought you hurt yourself in some way. But folks, this is really, really, to me, an epiphany. I think we have a tendency of looking the soil and the plant as separate units, and really they're really one unit. Because if you take the plant and soil and the microbe out, what do you have? When you take soil ecology, soil biology out, and the plant biology, you have geology. Or what would the other scientists call it? You have the geology. Boring. I mean, come on, geologists. But we need geologists, but I tell you what, I'm into the biology part. It's these two systems. This is the energy transformer why we are so critical, because it's these guys and the microbes provide the ecosystem services. Now, this is a slide I pulled out and created one out of that because a lot of people will talk about compaction and how about modification of the physical. Well, ladies and gentlemen, there are new evidence called niche construction. Evolutionist biologists they're having these big debates in the background, and they're saying, look, Darwin didn't always have it right. We used to think that if you put these organisms out into the habitat, they will adapt to that habitat. Well, that's not correct. That's only partial part of the story. The biology has the ability to modify the habitat. We know that beavers can modify stream systems. We know that bacteria and fungus and earthworms modify the soil ecosystem. They modify it. It's a two-way system because that's what makes the dynamic properties. It's the living soil ecology, the soil biology. They modify the system. I think this is a perfect picture of showing that modification. Peterson, why can those trees survive on this rock? What's going on? What do you think is going on? I'm going to call on you. That's why you have name tags.
Biology.
What is the biology doing to that rock?
The lichens have excreted acids that start to turn a rock into soil.
That and bacteria, they can dissolve rock. The mycorrhizae have the ability, these powerful enzymes, to excrete, and like Britt was talking about, it has the ability to bring up those nutrients from the mineral rock. Never forgot when first time I went to North Carolina, the soils were so destroyed with cotton and tobacco, cotton and tobacco, cotton and tobacco. Nothing could not even the scabs, the wheat could come out of that system. So the farmer said to me, the only way we were able to bring back those soils were through conifers. Why were conifers able to bring the soil back when nothing else could bring the system back? What do you think was going on, Bob? Why were the conifers so powerful? What did the conifers do to be able to extract phosphorus from the mineral rock? Erode it, substrate. They're heavily mycorrhizal. They were able to do that. And what I'm saying is, do not underestimate the power of the biology, and that's what we're really here about, is understanding that power of that biology. This is a 17-year soybean monoculture. This forest, systems right next to each other. It is, to make it look this way, it is the biology that does that. Niche construction, it can modify that. It can change that. What I want this is because then when you have a system like this, then that's why we use the term highly buffered. It can control these extremes in chemistry, climate, and temperature. This is why we want these dynamic properties, and it's driven by the plant and the biology. But the thing is that what we have in most of our cropping systems, either any part of the country will have wheat fallow, we'll have continuous wheat, we'll have diverse crop rotations, but these are the systems we want to see. And if we can stick them in between our corn and soybeans, what's the only thing missing here that I would love to see in more of this integration, Jeff? Because this is beautiful. I love that. Livestock. Now we're bringing ecological memory. Now we're bringing ecological memory into the system. So this is very important. That's our prairies, the way they look. I want the architecture on the bottom and the architecture on the top. I want to mimic that architecture. That's what we want. Because then we have redundancy in function, we have protection of the ecosystem, but we're also feeding different types of biology. This is how they biocommunicate to each other. The plants will communicate to the microbes. Now, that is what I want to see. That is beauty. That is what we're using these plants as like mobile links in these patches. You got to look at your field like a patch, that we're bringing ecological memory back. And when we build that, not do we only take care of the links on the bottom, but we take care of the links on the top. Then we bring the beneficials, we take care of that system. I'll never forget what my farmer friend, rancher Gabe Brown said. The problem with most farmers and ranchers is because they don't have faith in the system. They do not have faith in the system that they can self-regulate. These are self-regulating, self-healing systems. All we have to do is, in the basic, is start bringing some of the diversity back. That's why it's one of our principles. It's a hardcore principle. We want to create all these collaboration, these collaborative systems. They have done some radioisotope sulfur-32 and N15, and they actually, in these forest floors, they would cover these canopies, and they would follow the carbon from the atmosphere all the way to one part of the tree, and they were sending food carbon substrates to another tiny tree in that same forest. They were actually communicating to each other like Avatar. You know how sometimes we saw the movie Avatar and we're all laughing the trees communicate and they talk to each other. Well, you know what? That's not far-fetched. That's what natural ecosystems do. We have the technology now to follow those radioisotopes, and that's pretty darn cool. These plants are communicating to each other. See, for a long time, we didn't think that the Russians, I'm going to tell you some of the best soil microbiology books I have read were from the Russians. The Russians were way ahead. The father of soil microbiology was Wingradsky, wasn't it? And Dr. Kreskinov wrote a fantastic book called, I would definitely get this book. If you email me, well, here's what I'll do. I will send a list of my books, and I will send that PDF file of the soil microbiology book by him. It is the book I read very, very frequently. I go reading that book all the time. Ignore the first couple of chapters because it's a plating, but it's an extremely powerful book. What we want to do is build this collaborative processes. I'll never forget, in one of the books, is they had 2 pots with sand media. In one pot, they had just grass by itself. In the other pot, they had a legume and the grass. Where these two were put together, it leaked twice the amino acids when the legume was there versus the legume by itself. So when you put these two systems and you put a grass, it causes this legume to work harder. That's why when I do my lawn, I threw vetch and rake clover on my front yard. That's why now I rarely mow my front yard. I mow my yard 5 times and the neighbors mow theirs 30 times. The only problem with that with Jeff, is the neighbors look at me and says, Ray, as I'm walking around the neighborhood, they say, Ray, why don't you mow your lawn? I said, it's because I'm building a Rizosphere. The neighbor said, no, you're lazy. I said, get over, I'm putting goats and sheep on it. I said, luckily I'm zoned agriculture, guys, on my acre and a half, acre and a quarter. But I want to show you another thing that I want you to think about before I go into the Rick Haney test, is that how many understand the concept of fractals? How many have heard of the concept of fractals? Sean, tell us what fractals are. Well, at least tell us what you think you know what they know, or what you know about them. Because
It's a mathematical branching system.
Absolutely. Very good. See, he's been reading at night. He doesn't tell anybody. What are you doing under the covers? I'm reading fractals. Very powerful. In fact, I wish I could remember the Dutch scientist that came up with a mathematician. I think he was a physicist. They mocked him. His own peers mocked him and says, hey guys, there's mathematical patterns in nature, and they're mathematical. In fact, if you look at islands and how they're serrated, if you get closer down to the bottom, it's fractals. Trees, ladies and gentlemen, are fractals. Now here this is mycorrhizal. It's beautiful, that's a fractal. What is this a picture of? It's a long fractal. What is that? That's broccoli. Root system. Look at here, the arbuscular mycorrhizae. These are fractals. And the reason nature uses fractals, they're highly efficient. Very efficient for absorption and picking up nutrients, and also for the lungs. If you look at the end, now you guys need to help me in this because I'll mispronounce it really badly. At the end of those bronchial tubes, and they have these little sacs, what are they called? Alveoli, how do you say it? Am I saying it wrong? Help me out. Well, guess what? You know what I think they look at if you really look at them? They look like an aggregate. If you really look at them, they look like an aggregate. And right at that aggregate interface, a huge amount of biology happens in that process. So I'm saying is there's a lot of patterns you see in nature in wholes. That's why when I go out there, you start looking at nature in wholes and patterns. Now, this picture right here, a lot of you guys have already seen this, but I used this video. I tried to find it the other day, and I could not find it on YouTube, but I got this from YouTube. The moment we put those living covers, they start sending chemical messengers. They start communicating biochemically to these microbes. In fact, scientists think that as soon as you put that seed, that communication starts happening right away. So it's even before then. So what we're trying to do is trying to trigger more of this going on into that soil ecosystem. According to Dr. Pinto, his book on the Rizosphere, that root is coming down at 1400 psi. 1400 psi. This is the real food for the soil. It's the sugars, the proteins, the carbohydrates. We want to feed that, and we want to see that's called the carbon pathway. We want that carbon pathway because it's one of the major things to feed that soil ecosystem. And these are the grazers that are happening. These grazers, what they're doing too, as they're grazing, vitamins, antibiotics, all these things are being excreted, and the plant will take them up. When manure interfaces, you put manure and carbon substrates out there, and when the microbes work it, we have seen, the science has shown that the B vitamin concentrations increase in the seed head in wheat. That's why carbon is such an incredible, amazing thing. It's what the microbes do to the manure that makes manure awesome. It is what the microbes do to it. So I just want you to remember, that's why we're talking about, I call the root our leverage point. Root engineering is called rhizoengineering. What we're doing out there with those mixes, it's called rhizoengineering. There's literature out there on rhizoengineering where we can use certain metabolites, because Britt, I think was it you that were asking what kind of research is in the wave of the future? I think this is where we're heading, where we can use certain root exudates and say, look, if I need more phosphorus, we can put this part in the mix and this part in the mix, and we can get more phosphorus excreted. What we're noticing in everywhere, in Barry Fisher, who's a state agronomist in Indiana, he farms. Privately, he came and talked to me and he says, Ray, you know what we started noticing in my place? The phosphorus levels keep going up. And I'm not adding more phosphorus, but they keep going up. We're seeing this system happening. We're exciting that system. Please understand those covers are biological primers. We're making some of these substances more available. Now, people will confuse it and say, so, well, are we creating minerals, phosphorus out of nothing? No, we're not making magic here. The soil system has so much phosphorus in the system. I do not know, and I've been trying to scour the literature, I don't know how much available phosphorus. Some people say they do, some people say they don't. I don't know, it's up for debate. I really don't care. Now, here's the thing, what I do care is if you're starting to export huge amounts of phosphorus, you're going to have to give back. And what are some of the most extractive things we do in agriculture that extract nutrients, guys? Silage and one of our favorites, right, Doug? Haney, very extractive. So we got to think and take in that mass balance. But here's what we're talking about. Is some of these things we're going to be measuring indirectly, but this is some of the food substrates that those organisms are feeding. Amino acids, organic acids, sugars, vitamins, purines, nucleosides, enzymes, inorganic ions, gaseous molecules. Some of this stuff has been around. West, 1939. This is not new technology, guys. And I get so frustrated when I hear researchers say, but we need more research. We need more doing. We know a lot of this stuff. It's been around for a long time. Do I say not to stop research? No, that's not what I'm getting at. We need to take it we need to start applying some of this on the land. Now, here's a book I definitely recommend, but I'm going to tell you guys, it's not an easy read. It's about a $200 book. It's called The Rizosphere. And Dr. Pinto, his focus is, look, I've never heard some of these terms before until we started. Diffusiates, excretions, secretions, border cells, root debris. Doug Peterson's thought I make these stuff up. Or I was smoking too much cover crops and I came up with them on my own. But I've been accused of that. In my Peace Corps times, yes, I did inhale. Oh, no, don't rub that off, okay. But in seriousness, guys, look at the secretions, mucilus, protons, electrons, enzymes, cytospores, alleochemicals. Alleochemicals are negative messengers. There's positive communication and negative communication. The negative communications are chemicals where it's trying to suppress others. But let me give you an example. Walnuts. I used to think that the walnut chemical composition of the walnut shells were the ones that created jogaline. But it's what my understanding is when the walnut lands on top of the soil surface and comes in contact with the microbes, creates that chemical. Another one is Sudan grass. It's a Sudan or Milo, it's called sorgoline. That also creates oleochemicals. So I want you guys to be aware that when you're walking out in the field and you plant Sudan grass and then all of a sudden you come with corn behind, it can impact the corn. It can actually act like a natural herbicide and you can see a yield reduction. But it may only last 2 or 3 weeks in the soil ecosystem. Okay? So you guys need to understand that there's those chemical messengers out there and it can impact other of the other plants. But they're also food substrates. Just be aware that some of those things happen. There's a good example of that. In Dave Brandt's, this happened in North Dakota, but in Dave Brandt's, I'm telling you, our 8-10 wing mix, it was mind-boggling. It looked like a herbicide application was applied. When you could see the wheat, and it would stop, the voluntary wheat would stop right at the border where we had that 8 to 10 wing mix. It was amazing to see. And it's because of those chemicals being released and the byproducts actually suppress that voluntary wheat. So we can use this as an advantage for reducing herbicide applications. Okay, now, quickly, and then I will go, and this is part of my a PowerPoint I call, my series called the Plant and Soil 1, then I go into the Rick Haney test. Folks, this is what a microbial food web grassland looks is, and again, everything's connected, everything is one. One of the biggest things that we need to be aware of is these herbicide applications and these fungicides. And I don't know if it's happening here in Iowa, but certain parts of the area, they're saying if we, that if you put these fungicides out there, we get a yield increase in soybean. How many of you guys have heard that? Yeah. Well, that's what they say, and they're in Jeddah, and they've been going out there and they promote this ubiquitous spraying of this fungicides. Just recently published in the Science Daily, it came out from Europe where they did one field with fungicides sprayed and one field where they did not spray fungicides, and they examined both sides of the earth, examined the earthworms. The area where it was sprayed with fungicides, the earthworms were half the weight size. And if you know anything about earthworms, they have to be a certain weight before they can reproduce. So it was messing with their fecundity, sort of the spraying of the fungicides. The fungicides are not your friends. They are not host specific. They hurt the saprophytic fungus. They hurt the predator fungus, which also can help control nematodes. You can have nematode issues, and you can have phosphorus issues. You have to be very, very careful with your fungicides. So they'll go ahead and promote this, that you get the yield bump, but then you're sacrificing everything else. You have to be extremely careful what you do with fungicides, and you have to be very careful what you do with insecticides. So I'm very cautious. So if you take the mycorrhizae out, you affect the springtails, and you affect these critters, then the whole system collapses. Then you're in the vicious spiral I call, now you're buying fungicides, insecticides, and you're buying the fertilizer, and now you're this downward spiral. Now you have this addiction to this material. Let the system do itself. It's self-regulating, self-healing. What we got to do is provide the opportunity for it. Now, here's the instrument that we talked about. This is where we talked about yesterday what I want to see present. I want to see that present, this present, all these spheres of function, of areas of function. Spheres means areas of impact, areas where they're impacting the area. So I want you to think of this dimensionally. When we're doing the Rick Haney test, all these areas are contributing to the active carbon pull. In the Drizosphere, when you see earthworms pulling down, they bring the carbon down, and then they'll put it there for a stage, and then they'll let the bacteria work on it. And then the bacteria will break down, and those Drizosphere, the area of influence, is rich in huge species of bacteria, high in phosphorus, high in nitrogen. That area impacts the rest of it. What we're saying, all this area impacts each other. They all contribute to the active carbon pull. The aggregate, you see these grain sand particles? They're fused by these organomineral complexes. This is the house that lasts 27 days. The system is always being fed on that. We have to replace that with living covers. The Rizosphere, you can't have this. This is the conduit of energy that feeds this, feeds this. All of it's connected. I can't have this pores without this. This keeps it intact. So all of it is important. The Detritus, that shelters the soil, protects it from the shelter of the rain impact, but also the grinders and the mixers, they will decompose and all that material will leak into the system. So all of it's connected. This is the aggregation that I like to see. I call my cottage cheese, or that granular structure. This is Gabe Brown's soil. What was it? They had a 12-inch rain in 24 hours. It doesn't look like mush. These glues are hydrophobic. They shed water, okay? And there's also a food substrate. This is a great picture showing what happens when you create aggregates. This is a very sandy soil. Sandy soils will be, in the upper 1 inch, they can aggregate. See that surface of that sand particle? That whole area was coated with those organomineral complexes. Now we start to have cation exchange capacity. Now you help regulate temperature. Now you help regulate pH. Now you have helps with nature potential, electrical conductivity. Once you bring these aggregate, you build organic matter, what is the best way to remediate salinity? Sean, Eric, what is the best way to help? When we have a saline sodic area, you don't have that here very frequently, but when we have this in the West, you know what we do to remediate saline sodic soils? Gypsum. We usually use gypsum. That's not the way to do it. If you have it in an area where it's high pH, we use sulfur. What else do we use? To release calcium off the exchange site. We want to knock the sodiums off the exchange site. What we do is manure, cover crops, stop tilling the sucker. Why? Because if we build these aggregate, we can help regulate this stuff. And it has the ability to ameliorate it, to keep it out, occlude it. Right now, some scientists say that some of this supermolecules in organic matter have memory, where it can actually exclude the salts and some of these toxins away from the plant. We are just beginning to scratch the surface. But if you cannot maintain an agrosphere, you can't get the rest of this stuff. This is very, very critical. Very, very critical. For regulation, all this. This is the habitat that we're wanting to regulate. Okay, now, why are we gotten away from this model? This is the model that I was, as an agronomist, trained. The inorganic-based soluble state model. This is what was pushed on me. That when you, okay, now how does this soluble inorganic state work? Guys in the back. How does this model work? What was it? Diffusion, mass flow, and what's the other one, Jeff? Remember that? In college we were talking about? Interception. We were pushed at, that's the model. Problem with that model is, guess what? We're losing 40 to 60% of our N. We're losing 60 to 40%. We have soils that are bare 4 to 8 months, and what we've done is we've decoupled the carbon and nitrogen and phosphorus cycle. Does that happen? Yes, it does happen, but that's not the right paradigm to look upon it. The right paradigm is this. Dr. Drinkwater from Cornell University, Dr. Michael Swift, some other scientists are challenging that. They're saying, no, our system is ecologically based and it's organo-mineral pools driven, microbially plant mediated process that drives the inorganic. This drives that. They are together. So when you have this kind of paradigm, then your strategy changes. This is why the plants become such a critical process. This is the model that we're addressing. Now we have, here's where the Haney test hopes to pick some of this model, and we've been missing this part in our equation. So that when we've been doing the university soil samples, we've been focusing on this and we've been missing it by a mile. And it tells us go put more fertilizer. So we're saying no, put it all together. Look at the system as a whole. What percentage of our fertilizer actually reaches the plant? Class? Anybody have an estimate? Jason? If we put fertilizer out there, how much actual of the chemical fertilizer could they done radioisotopes? And if you can find it on page, if the nature and properties on the 700, 720, 730, 740, somewhere there, it talks about how they did radioisotopes and they followed it up. What percentage of the chemical nitrogen actually reached the plant?
40%.
Yeah. 40 to 50%. Where did the rest of the nitrogen go? Organo-mineral complexes, some of it leached, some of it, but it didn't reach there. So what we're saying is, this how much percentage, whether you're in continuous corn or soybean, a majority of it still came from the soil. And that's what farmers get confused. They actually think that the chemical fertilizer is providing all that nitrogen, and they're expecting all that efficiency. Here's the new research. It just was published recently in Science Daily, Science Daily showed it, but it was really a paper from Europe, or I can't remember where it was exactly. But the beauty about it is they did radioisotopes and they found out, oh, look, the mycorrhizae are taking up organic molecules, amino acids, into the hyphae. I mean, they're sucking it up into the mycorrhizae and taking it into the plants. And they said, whoa, that's cool. The Russians knew that since the 30s. That plants have the ability to take up organic nitrogen through amino acids. Amino acids have a nitrogen in it. Plants cannot just only take ammonium and nitrate. They can take up organic molecules also. Amino acids inside the mycorrhizae. They take them and they put them right into the plant, and they have the ability to process them. So when you're growing those covers right next to them and they're leaking that is equated. Also, do we also equate for predation? When protozoa and nematodes are eating bacteria, they also release ammonium, anywhere from 16 to 116 pounds of N. Do we take predation into consideration? So that's why I come to the conclusion 3 years ago, nitrogen is not the limiting nutrient in the soil. It's carbon. Why would I say carbon is the limiting nutrient? Guys in the back, what do you think? Why is carbon, why would I say carbon is the limiting nutrient? Mark? Are our soils carbon depleted at this point? Everywhere you go in this country, the soils are carbon depleted. They're not at the stage that they were. They're the drivers. Mark, you're exactly right. They're the drivers. Here's also some electron microscope images of fungus holding onto calcium crystals. I never thought that would happen. Okay, this is how we look at priming the soil. This comes from various sources. There's destructive priming and there's constructive priming. I want to focus on constructive priming. Jeff, how is this prime the soil? You know, everybody know what I mean by priming the soil? Release the nutrients. You can release, I had an old rancher tell me, he said, Ray, I am not going to start using the disc. I get a green up every time I flash. And I said, yes, you burned the house down to warm up a hot dog. Well, you should have turned the dang grill on. How does that prime the soil? You should be able to explain this.
Inject oxygen into the soil.
How?
Inject oxygen into the soil.
Okay, you inject the oxygen, and what does that do?
Wakes up
Our strategists, the cryptotrophic, they go crazy, they eat the carbon substrates, release CO2, release nitrogen, boom, bang, you got your nitrogen release, right? Then they got die-off. How does commercial fertilizer do that? Direct nitrogen source, right? The salt, feed it. Did you guys read the recent research came out from Illinois about what fertilizer does to the soil system? They've been arguing back and forth for debate. And I'm not going to go down that road, but to tell you what, I tell people use that substance with incredible care. Fire, big debate about fire. What's your guys' take on fire and how it releases and primes the soil? Peterson, what do you think?
I think it's sort of like a tillage. It's a quick release, but long term, it's typically, in most cases, it's going to be degrading.
How about you, Brett? What do you think?
Well, fire was a part of this ecosystem before people were putting crops on it.
Yes, they were.
Kind of acted to reset the system, kind of get out some of the bad plants and promote some of the plants that were more natural part of the environment. So I don't know that fire should necessarily be considered always destructive.
And see, I think Doug and all of us agree on that. See, we're talking about these very, very hot fires that can actually create, you can actually get so hot that you can actually create the sand particles to come into an amorphous substance. You can actually, like glass, fuse it together. You can burn off organic matter. You can volatilize it. You can seal the soil surface. It can be damaging. If it's done a cool fire.
We think you have to have a good burn, and that's probably the opposite. You need a
Yeah.
spotty,
Yeah.
effective burn to do the most biological.
Yeah, as you have to be very careful with your fire. You have to be very careful. I think it's the exception, not the rule. It was the buffalo going up, down, not the fires. It did happen. You could see the benefits of it, but you have to be careful with it. If you got a lot of fuel and you get really hot, you're going to have some issues. And the literature backs that up. They've seen it and they've documented it. You can have erosion, you can have issues, you're going to lose organic matter, you are going to lose carbon. You're going to lose that. I don't think but guess what? Our farmers in agriculture, like Idaho, where I farmed, what did they do? They burned all the darn residue. And you had these huge plumes of carbon going into the system. You just burned 10, 20% of your soil energy up in flames. Any other comments about the fire, you guys think?
See, I was going to actually mention too, is this whole argument about destructive fires. What are you thinking about? I mean, is it a perennial system, or is it a burst system? Or
Yeah.
is it an annual cropping system? I mean, there's two different systems here, that one is completely destructive and the other is potentially beneficial.
I love Jeff. Context. I think that's probably one of the most critical things, knowing your context ecologically, what's going on. We have farmers in fields that are just lighting the match through because they don't want to deal with the residue. And that's still happening, guys, in Washington State, in various places. But you just need to be aware of that. Now, here's my constructive release of nutrients. Now, I did put fallow here, and it's interesting. I put fallow, and I don't know if it's really a priming system, but what they do is they store water. Why do we have fallow in some of these dry areas? Just to store water. But the reason I put that there is because it's interesting. In these fallow systems, we are not building organic matter. I've been to Pendleton. I've been to some of the research centers over there at Oregon State and that. They get to a certain level and they can never go any higher in building organic matter because they have the whole summer they store and they gain no water. They're just storing water. So these systems always are stagnant, whether it's no-till or conventional, they're stagnant. And we're arguing to them and says, look, we cannot afford to keep that bare. But it's the water issue, so how are we going to build a bigger bucket if you can't build it with the plants? You can only build a bigger bucket if you have the plants. And we all know that even fifth graders know plants suck up water. They need water. But do we want our water to go through evapotranspiration or to evaporate? How do we want our water to be used? So that's the thing we have to think about. Root exudates, primer. That's the primer. I prime this, all these organisms come. Then the party begins. Manure, compost, and anything that's carbon-based, this is the way I want to prime my system. That's the way I want to prime it. Again, to me, that's the main one right there, carbon. That's the keystone. Look at here, control soil, NP and K, raw manure, composted manure. What happened here with NP and K? Why does the root system look different? Eric? Yeah. Control soil, chem, raw, compost.
No biology to make it available.
And what else do you think is going on?
Well, if it's only in the top surface or a very limited location where your NPK is available, then maybe it only went where it was.
What is the salt context on some of these fertilizers like urea and them? Have you ever seen the salt context? So, the salts in chemical fertilizer. Anhydrous is huge. Urea, high. Salt's brutal to the system. You have to think about, they are salts. You have to be careful with our tools. Okay, Gabe, I'm going to go through this quick. This is where I started using, right here. It's where we started questioning, I started questioning some of the stuff about 5, 6 years ago. I started questioning how we were doing, not only how we were farming, how we're taking our soil tests. It's this man's operation that started getting me to question things. Gabe, Jay, and all these guys started questioning things a long time ago, especially when it came to our soil samples, many, many ways. But again, give you a little background about Gabe real quick. 15, 16-inch precept. 3 to 5,000 acres, these farms, about 2,000 cropping, 3,000 range. He has completely eliminated inorganic nitrogen and phosphorus in operation. He wants to complete, he is not a fan of Roundup. He wants that completely away from his operation, period. In fact, I don't even know if he uses it anymore, period. He uses some herbicide. He is not an organic guy. I don't care about organic, but he does care about the resource. Both of us, Doug, Peterson, myself, Gail, all of us talk to each other on a frequent basis. We're trying to go figure out how we can go to organic no-till. No input, no-till. That's what I mean. In other words, using petroleum, no petroleum base. So we're trying to figure out how we can do the right rotation so we can do that. He has been, that man will never be satisfied. He's always pushing the envelope. Here's what happened. We started questioning things. 62 bushel dryland wheat. With a 16-inch rain, he can get 62 bushels, and the average yield in Burleigh County is 35 bushels with no fertility. How does he do that? How is he doing that? If we do a traditional soil test from the university, you go in, he comes into the office, we would say we need this much nitrogen, this much phosphorus. This is the traditional approach, okay? And then if you do it with the university soil lab, not the private lab, his soil sample showed he only had 10 pounds of N, and 6 parts per million phosphorus. Is that enough to grow 62 bushel wheat? No, not even close. But the university soil samples told us we need to put that. And we started seeing that. I started seeing that in Pennsylvania. I started seeing that everywhere. But I was not aware of it until, especially when you start getting into no-till and these healthy soil ecosystems, I said, this tool is not working for us. In fact, Gabe calls these tests, his son calls them the piss test, because you piss fertilizer away. It's not until we started coming with a Haney test that we started getting closer of what, and I'm going to get into that, why there's a big difference between that test. How is he doing it? No-till, 10 years, diversity, cover crop, ultra high stock grazing. Results, no fertilizer. Okay? That's what he's doing and how he's doing it. He's using carbon and nitrogen from these sources. These are legume mixes, gaining 2 to 3 pounds per day. Look at the manure distribution on that. Okay? In his place, we'll go from cropland with no livestock, cropland with livestock, and then the native range is in the 7s to the 8s. He will tell you, but he started at 1 to 2%, and he's already pushed that system that far. Here's a young man that this is the gentleman that watched our videos. He's in North Carolina. He watched our videos, undercover farmer. He's a fireman, and he went and took some forest land and pasture and converted it. He had soil health scores of 10 to 12. A soil health score from the Haney test from 7 and above is you're heading the right direction. When you're 7 below, you have some issues. How did this young man take a pasture and a forest to a multi-species cover crop and then plant corn and come up with 230 bushel corn? In fact, you can listen to his story on the webinar that was just done a couple of days ago. This is the young man. I definitely, you'll find him in our East National Technology Service webinar. I want you to notice is his check strip. This is the way his cereal rye looks now in March, in April. It's already 36 inches high. This was planted in the fall. Do you know what this is? It's a check strip. This is the way the fields look typically. Do you know what that is? Is a weed. A little weed that comes out. That's the way it normally looks in North Carolina at that time. This is the way his looks. It's called, he calls this the Holcomb 5-way blend. This young man already picks that and says, well, Russ, what do you want to do now that you're catching on to this? I don't want to be a fireman anymore. I want to farm full time. So that's his goal. But he knows this young man's already a believer. We're going to go to his field day and we're going to showcase his cover crops. Now, The reason I start you off with that, give you some context, this is the Rick Haney test. It's gone to a 20 peer review. It's been around for a while. I met Rick Haney about 4 or 5 years ago in Texas. He was one of the speakers, and I'm going to tell you, Rick Haney was not an advocate of no-till. Dr. Haney was not an advocate of no-till. He thought no-till was chemical farming. And he said another thing that intrinsically bothered him when you went, and he said, it bothered me when I'd go spray, but I'd have nothing growing. He really believes deep inside, intrinsically in all farmers, we love to see things growing. So that's why he had a problem with no-till. Too much chemicals and not growing enough and doing that chem fallow. So that he had those issues with it. After he sat and listened to a talk that we talked about how we promoted no-till in the covers, he picked on it real quickly. Now half of that ARS station is no-till with covers and no-till conventional with covers. So he's already in his third year now of research on, and we're collecting data on how the covers are doing and how they're changing the biology. If you ask Rick, if we would do one thing that's be very powerful, all agriculture, even if we're still tilling, is to plant a cover. He's seen soil health scores still raise with even tillage. The covers make that huge of a difference. Now with no-till, and I cannot wean you off some of these heavy nitrogen inputs unless you're no-tilling. And I will tell farmers that. So and Rick Haney is an advocate of no-till, but he'll tell you if we can put covers into the system, it would make a huge, huge difference in his opinion. Now, here's what Dr. Will Brinton, he did this little graph for us because we're getting challenges. I mean, let me tell you something. There's this thing going on with some of the state labs or some of the universities' soil testing labs, because there's this concern with the Haney test, and the concern is that one, they've talked about, well, it's a new test and where has it been calibrated for yields? Well, the argument is also how they've been calibrated. The regular university soil samples haven't been calibrated either. If you really look at their calibration, they've missed the organic fraction for a long, long time. So we are in the process of calibrating this with yield. And what I like about this test is the reason we did this table is to show you the different ways these extractants are done. The Haney test is based on the H3A. It mimics the soil environment. It uses the soil's natural extractants, water and root exudates. I got excited about this right away because I'm a huge advocate of biomimicry. One of the top soil scientists that told Rick Haney, his mentor, or he studied under, if you want an awesome soil test, use a test that disturbs the soil the least and not force the soil to say something it's not telling you. I love this test because it uses water. What does the soil see? Soil water. What else does it see? Root exudates. What Rick did is he searched the literature extensively, and he picked 3 major extractants that he found that roots give in most plants. So the root exudates are citric oleics and lithium citrates. This is what this is based on. This test adjusts to the pH and extracts nutrients like plants do. It adjusts to the soil, the plant, and the micro-mediated process. The disadvantage, it's new to the lab, and it's really hard. It takes a lot more time to run, okay? The break test, it uses strong chemicals. Pulls P out of the various complexes. It reacts to bond phosphate. It's acidic, got acid, it's toxic. There's some disadvantage to it. Same thing with the malic. These things are forcing the system and forcing things out of the system, and I don't think this is old technology. This technology has been in the 30s, 40s, 50s, 60s, for the 40s, 50s, and 60s. There's a reason why it's called Malik 3. Malik's been working it for a while. He had a Malik 1, he had a Malik 2. Now it's a Malik 3. Before he died, it would have probably been a Malik 4. Morgan's, strong chemicals, buffered sodium, all these chemicals. What does the natural system see? Water and exudates. Europe uses mimic soil iconic strength. This is what they use. Not recognized in the US. Adjust to soil, safe, 40 years, EU experience. What I like about this test, it gets us closer. We've already done 4,000 to 6,000 samples all over the country. We have saved anywhere from $20 to $30 per acre for farmers. We have figured we have saved $20 to $30 million at least in cost for farmers by doing these soil tests all over the country. Huge difference. I had one farmer in Ohio save $60,000 by following the Rick Haney test. Okay? This soil sample is taken like a regular university soil sample. I will have this PowerPoint for you guys if you want it, okay? You make sure you'll get this. Now, Rick Haney has been personally mentoring me, and since I'm a slow learner, we've been going over and he's been teaching me this test over and over, and I've had to help. He's taught me how to teach this. Every time I hear that man speak, I learn something new. So that's the way learning is. Here's how that's based. I want you guys to think about, this is College Station, Texas, Lubbock, Amarillo. Okay, soils work like this. You have dry cycles, wetting cycles. Since soils are aquatic ecosystems, if you imagine you have a dry cycle, the water comes into the soil ecosystem, all of a sudden, again, it's a subaquatic system, all these carbon constituents, dead microbodies, carbon, all of a sudden, the system starts becoming more active. The higher the fertility, the higher that CO2 respiration that first day. Rick did not come up with this. This has been around since the turn of the century. Scientists have been talking about this for a long time. Lubbock, this is less fertile. This field soil is less fertile than this one. So the higher the CO2 respiration, the higher the fertility. The Rick Haney test that's only one parameter of the Rick Haney test. The concept is here, you're having all these carbon constituents, these byproducts of decomposition happening, and they're leaking into this system. The soil system is an open circulatory system. It's like an engine, like the splash mechanism. Have you guys ever seen how an engine inside, how it splashes oil? We're a closed circulatory system. The soil ecosystem is an open circulatory system. So all the carbon constituents, all the food, everything. A soil scientist, I can't pronounce his name, from Russia, he considered the soil solution like the blood of the soil. When you go to the doctor will sample you, give a blood test. Why does he do a blood test, Britt? Why would he do a blood test on you? What is blood test tell us in the human body? I can know I can tell about liver function. I can tell about, what else? Cholesterol. I can tell all kinds of cool things just with that blood sample. The concept is the same. The soil scientists really believed that the soil solution was like the blood of the soil. So what's happening is we're picking up all these organic molecules and we're able to re-wet and dry the system, and then we're picking up those active carbon constituents in that system. So this whole process is happening. So keep in mind, you're bringing in the Rizosphere, the roots, all that, superimpose all that system contributes to this system. It's subaquatic because some of it's submerged underwater and some of it's not, but it's an aquatic ecosystem, okay? So here's that layer I was telling you about, the aggregation zone. All that will become part of this whole system. These critters, when I want these systems, these critters need to be present for this to happen. Okay, here we go. You can have a massive house I had one of the soil scientists in California say, Ray, well, what do I do with these guys? How am I going to help them with soil health? They got 10% organic matter. How are we going to help them out? I said, well, are their soils cycling on their own? If I took away the chemical fertilizer, would they still function? Here's the point. You can have 10 or 12% organic matter. You can have a huge house, but you can still be eating hot dogs every day. It's the quality of carbon. This house is the carbon that can no longer be broken down. The only way you can break this other part of the carbon down is how? How can we get on that carbon that can no longer be processed? How can this be broken down more? Burned. Or you put more carbon and then you can use it as a priming to break more of it down, but it's really tough. That's pretty rich to a stable state. We want this once. This is what the organisms are focusing on. This pool is 80 times smaller. It is the carbon in the water extract. All of Rick's tests, analyzing machines, pick up things in the water extract. Ingenious. He re-wets those soils back, and he's able to pick up some of this. These analyzers are able to pick up the nitrogen in the water solution, the carbon in the water solution. It is the food they eat that we're looking at this. This test focuses on the food. This is where we've been missing. We've done on average of 6,227 soil samples. We have missed this much of that system. If we were to look at that, we've been missing close to half of the nitrogen. And we could see it in the field, but we could not explain it. This makes me feel more comfortable where we're at. Dr. Haney's wife, he married a 35-year-old, his wife's 35 years old. Rick's 55 years old. He's got 2 little kids. One's 2 years old and 9 months. I said, Rick, how in the world did you manage marrying such a young, nice-looking woman, besides has a master's in science and going to a PhD? And he said, cover crops, Ray. Cover crops, man. No, I'm just being funny. He says, Liz Haney made a comment. He says, by the way, if I said that, farmers will be signing up for cover crops. No, I'm just kidding. His wife made an interesting comment. If plants weren't able to bring up organic compounds, herbicides wouldn't work. Herbicides wouldn't work. Plants were able to pick up organic molecules for a long, long time. We've known that. Here are the machines, the sophisticated machinery that when you send the soil sample, this is what it said. That's why it only takes 200 or 300 soil samples. A lab can only run 200 or 300 soil samples. Now, I want you to keep in mind, we're only going to do Iowas or South Dakotas. Everybody's signed up. You guys all know Dave Lamp has been sending out mass information. We are going to get select landowners that are going to work with us and share information with us. We're going to run the soil samples through the Rick Haney. Everybody else is going to have to pay for their soil sample. Okay? Rick, the gatekeeper here in Iowa is our beloved Rick. Rick and Barb are going to be working together and making sure that they're the ones that are the gatekeepers. We cannot do everybody's soil sample. NRCS gave Rick Haney $100,000. We gave him $100,000 so we can test this. We're going to run more, calibrate, and continue working these tests. But we need information. We're going to need yield. We're going to need some of the information so that we can continue to build our confidence in this test. Okay? That's the concept. And you're going to be the gatekeeper over there.
Actually, it's Eric Barson's our agronomist.
Oh, Eric, okay. It's okay. So you guys are going to work together, but remember, everybody else is going to have to pay. Now, there are 3 labs that do this test. Woods End, Ray Ward, and what's the other one? Doug, do you guys remember anybody else? I'll remember. There's 3 of them. I think it's at the end. But again, here's the things that this Rick Haney sample does, and they run some things on the background. And we've been very careful that these three labs are speaking very intimately with Rick Haney, okay? We're all on the same sheet of music. The soil health calculation. If your soil health calculation is less than 7, you have some soil issues. You have issues you need to address. Your soil is not there. 7 and above. Gabe's Brown came back at 18 to 21 to give you a perspective. He has one of the higher soil health scores in the country.
How high does it go?
Up to 50. Now, here's the thing. Extractable carbon, this measures extractable carbon in the water solution. Percent MAC is the microbial activity and how they process the quality of the carbon and what the microbes are doing to that. Extractable organic N, inorganic N and P, organic carbon and phosphorus, the ratios of that, which is very critical, organic N and P ratio. And of course the Solvita. It uses all those parameters. It doesn't just use one parameter. It uses all those parameters to come up with a soil health calculation. It uses all the parameters. Here's what we're talking about, the difference between the burst test and the basal field test. What we did is the basal field test. When, Rick, you send the soil sample back, he will use a certain amount of water, and it's measured. He sticks the paddle out there, and through capillary action and through gravity, it will re-wet by itself. That's the difference between the burst test and the basal test. This is what we did. This is what I'll do when I go out there with my 3-inch. This is what I'll do. Okay? Again, you can find all the information on the Solvita website. I have the meter here. This meter is $1,400 to $1,600. Okay? I got one for the tech center. Rick's got one. So I don't know how many do you have here, Rick?
I have 2.
You have 2 of them, okay? It's very easy to do. You turn the meter on, split caliper, and then you follow the instruction. You stick the whole paddle in, push read, and it will give you a digital reading, okay? I did the no-till. The color's already turned on this. I did the no-till earlier this morning. The no-till was 2.95 with cover, and the till was 1.9. And I'll show you the color in a little while. What does that mean? Here's another thing that would work pretty good. See how you put that little tube and then you put it back in? I think that would work pretty good. Have you had that one, Doug? Do you have that little tool right there? And see, if you look at it, the color is correlated with parts per million CO2 released. Here's the thing. I told you it was 2.95. The higher the color, the higher CO2, the more it relates to N mineralization. The lower the colors this way, the lower the activity. Now, here's the thing that's very interesting. I have seen very low activity soils, but have huge amounts of carbon. What do you think that's telling you? Huge amounts of carbon, but very little respiration. What kind of... It could be CRP land that hadn't been grazed for a long, long time. We've seen that. Very little activity. Huge amounts of carbon in the system. It's not being processed. Very good. Thinking like a microbes. There again, impoverished soil, borderline, worsening, transition into good, extremely vibrant soils. The farmer I work with, Ray Stiers, eliminate fertilizer, he's right here. Okay? And I'll show you a little bit here. Now, well, that impacts it. Very good. Good. Thomas is saying is when you take these samples, make sure you take them in the same time periods because that will help. That would impact it.
So, I mean, today's 2.9, that's...
Yeah. Yours was done yesterday, right? Yeah. And you got, good point. So but here's the thing when I want you guys to think about the tool. When would you use the tool? When you want to find something out. When do farmers want to use the tool the most important time? Is when they're going to go spend thousands of dollars out there and they want to know where they're at fertility-wise. Thanks, Tom. That's true. You always got to remember that. Context, context, context. Always remember how you're using your tools and what the information you're going to get from it.
Ray,
Yes?
you said earlier that Gabe Brown had the highest, about 18 to 21 on
Yeah.
the soil rate. Has anyone ever done any native prairie to find out what that is?
I have, yeah, in fact, I'll show you one. Very good. Okay, so one of the things that happens a lot of the confusion is that sometimes the farmer's saying, when you have green manures or covers, sometimes they focus on feeding plants. Sometimes farmers think that, again, chemical fertilizer and all that is what feeds the plant, but really what obscures that whole thought process, they forget that it's the plants and the soils that really feed the soils. And we have to remember that. It's the plant that feeds that soil ecosystem, and it's the microbes together. So that's really important to remember that when you're talking to farmers. Again, the way the system is set up. Here's again, Rick. This was taken in Vibora, not Vibora, I'm sorry, Kuchera. This is on his farm. Kuchera has a 2-3 acre field that has never ever been, it's been a native prairie as long as he's known and as long as they've owned that farm, it's been a native prairie. That was taken out of his place. Look at a no-till on Ray Steyer's, a virgin prairie Nebraska soil, multi-species cover crop, no-till, bare. That was taken last year. I took those soil samples. Here's, yes?
Sorry, is that grazed?
No, it wasn't grazed. It was just mowed. I don't think they grazed it. I could be wrong. I don't remember, but he said they never did anything to that place. I don't think it was grazed because it was just kind of a 2-3 acre obscure little field. Okay? And that would make a difference, make a big difference. But here's the thing, coming back again, in the slant test for soil liable N, that virgin prairie only had 218, but Ray Steyer's had 278 parts per million. Ray's soils, that's why his soils, if you sample his soils, they have like 150 to 180 units of N. He never applies fertilizer. But he puts cereal rye and crimson clover and veg 7-way mix for 30 years. He's got 6% organic matter on the upper 3 inches. You can do it. You can. Now, when we do our soil samples, this is taken right out of our, everybody has the soil, the kit manual. Okay? All of us have that. You're going to take the Haney test like a regular soil sample. You can isolate certain spots, sandy soils, clay soils. The more composite samples you take, the better. So in other words, I would take a 5-gallon bucket, take about 20 to 30 cores. The more cores I take and you mix it, I got one sample. You send a couple of pounds, you send it to Rick. So the more cores you're going to take and you mix it, the better your sample's going to be. Yes, ma'am.
So you're saying you would take 30 cores over a 30-acre field? And
Yeah, or 30 or 40, depends on you, yeah.
Okay.
And then if you got a, say you got a 40-acre field, but say 10 acres of it is clay and the rest of it's sandy, I'd isolate it. Treat them separately. Okay? Now also too, if you have a trouble spot and the rest of it's low, I would use that same concept. Don't take, and then what's some of the rules we do when we take soil samples? Don't take it by where? What's some of the no-nos for taking soil samples?
Right to the road, the field edge, right, the old feedlot.
Yes. Use that commonsensical thing because your numbers are going to be screwed up. So just be aware of that. Like trouble spots, like again, that's what Jeff was talking about. You got to be careful the way you take your soil samples. Here's what you get when you get woods in the laboratory. That's how the soil sample analysis looks like. He uses these beautiful pie charts. When you do his test, it's $55. He uses the university soil sample plus the Haney Britain test. You'll get both of them. It's $55. So you'll get water extractable carbon, the ammonia and phosphate, calcium, iron. You'll get all of these. And in fact, it's changed now. In fact, it even looks better in this, and it gives you a soil health score. This is an older one. Here it is. Soil health reading, organic carbon-nitrate ratio, aggregate stability, microbial active carbon. And he also now has a little box. It says you need to put your mix 50% legumes or 50% grass, 50% legumes. His soil sample will say that, just like the Haney will. But it's more graphical, easier to read, and it incorporates the university soil sample too. Okay? So you're going to have 2 soil samples in one. $55 for the soil sample.
For
Yes.
Haney or?
For Woods End.
Okay.
It's the Haney test. But when you go there, you're going to say, I want the Haney test and the regular soil sample. If you do the Haney thing, I think it's $30 by itself. 20 or 30. Don't hold me to that. But if you get his for $55, you get all of it. Okay?
Is Barney's the same way? Ward Labs?
No, Words Now is not the same. Yeah, no. We're going to try, I hope that maybe, I like what Will did with his. He put a lot of time and thought process to this. It's easy to look at, easy to read. I like that. And I don't know how far Ray's going to go with his. I don't know how Ray's looks like.
So far, we've sent in an array, what you end up with is a spreadsheet for the Haney test.
Oh,
If
okay.
you want a conventional soil test run too, you're gonna have to
Yeah.
pay for it.
Yeah. And how much is, I think he's charging $40-some, right?
Well, yeah, but he's given, at least us in South Dakota, 30% rate, so.
Okay. But when you go to Will's, you get this nice readout. And I really like that. I think Will's done a really good job, but here's what's going on. Will and the other labs, they're intimately, we're doing quality control. They got to be saying the same thing. They got to be saying the same thing. I now do not tell farmers we're doing free soil samples. I only do for the ones that the gatekeepers allow. We can't afford to do this because he's being overwhelmed. And the ARS was getting concerned. He was running a lab. But when the director found out what we're doing, he had no problem with that. This is research, but we're part of that research. Okay? This is how it looks now. This is the new, I thought I put it up here. This was done, the race tires. This is the upper 3 inches. And it'll tell him the nitrogen requirement for 150 bushel corn, no nitrogen, no phosphorus, no potash, none. Mix recommendation, 100% grass. Look at his nitrates levels, medium, high. You got all these water extractable carbon, water extractable nitrogen. His soil health score is 22.5. Organic carbon nitrogen ratio, Solvita. It gives all this, the aggregate stability. He'll run aggregate stability for you. You got everything in that one test for $55. He's got, and then it'll tell you in this pie chart what a majority of the nitrogen species is in the ammoniacal N. It'll tell you if it's in the nitrogen N. And the calcium versus the other ions and the microbial carbon, what the fraction's in. So it runs all this for you. And the percent organic matter, like the regular university would give you, his Ray Steyer's is 6.9 on the top 2 or 3 inches. You've been to Ray Steyer's place. Okay?
So we looking at it, supposed to be doing a 3-inch sample, or traditionally we do a 6-inch?
6-inch sample. This one
This is 3.
I did 3 on purpose.
Okay.
I wanted to see, I did this because Will was paying for the class. Will was paying for the test. We wanted to see what's in the top 3 inches, and we wanted to see what's in the below the other 3 inches.
His overall score, you would expect to be higher, I guess, on the 3.
Yes. Now, when I did the other 3 inches, what it dropped down to, the organic matter dropped down to 2. So you take the 6.2 and the 2, realistic is 4 or 5% organic matter, and that's pretty dang high. Because if you really look at the forest, but before Europeans entered in North Carolina, we're 3% plus. He's doing some darn good job on his soils. But again, he pumps a lot of carbon into that system. Now, remember that soil when I first showed you guys, the one that blows up last night you saw? That's the soil test on this one. 5.3, you can see the huge difference. Look at the aggregate stability, low. Look at the requirements, nitrate, nitrogen, low, everything's in the low. So these are those 2 soils that I use on my slate test, comparing side by side. And you can see the difference microbially, everything. Okay? I like what Rick did here with this. And They're color coded. It's a really nice table, easy to read out. I think he's done a really good job. Yes, sir.
Is the recommendation 100% grass because everything is so good that he can get by with just doing grass this?
Yeah, because he's got so much nitrogen in the system, but I'm going to, great question, more context. You have to use critical thinking too. If I'm going to go to corn but he's doing such a good system and he has so much nitrogen in the system, I may still want to go run the test to see where I am in that part of the spring. We took that test. When did I take that test? I took this test in the fall, okay? I may want to take it in the spring and see where I'm at. It depends, your timing, where you're at, and I might not necessarily say, I may still want to put 4 or 5 species out there, okay? It might not necessarily be grass. But if I'm doing with a guy that's absolutely degraded and he tells you that you're in your context, he's in early stages, I'll put some more legume mixes. And the test will tell you that. But you still have to do some critical thinking. How about grazing? Are you going to graze it? Maybe I want some legumes. Maybe I don't want, you got to still do those kind of thought process too. This is one answer. Only one part of your tool, guys. You have to do the other thinking too. What does the farmer want to do? Maybe you can't grow this. You have to bring all that into play.
How quick did you get this back?
Pretty darn quick. Well, this was done.
Court dates.
Yeah. What date was it?
February 14, and
February.
it says they received it in June.
8 months?
Yeah.
Yeah, I wouldn't worry about it. It was quick turnover, but I don't remember. But it was. You'll get him in a week or two. It's pretty quick. Now remember, this lab has just recently started. This lab's up to speed. Rick Haney and myself talked Will to do this. I said, Will, we need another lab. And he's located in Maine. So he's just recently geared up. This is fresh, hot off the press, guys. So he created this setup.
What's the difference between the right and the left? Are there 2 different tests?
This one is a no-till, yeah. This is zero no-till, rolled cover crops. This is the vegetable farm, right next to the same soil type. Cecil Sandy clay loam, Cecil Sandy clay loam. They're a mile apart. This is heavily tilled, vegetable, never covered. This is covered 24/7. He has completely limited nitrogen and phosphorus in operation. He's down from 5 herbicides down to 1 herbicide.
How do you know what P and K tests they don't see it listed anywhere?
The P and K test, they are based on the aluminum iron ratios, okay?
So I thought they were doing also the traditional tests like 3.
Yeah. I don't know where that one's infused in that one. All I know is when we do the Haney test, they use the aluminum and iron ratios on that. And the reason Rick Haney uses that, because he feels that's more accurate for pH and for some of those species of ions. So the potassium and the phosphorus, the extract for the Haney test is the root exudates, okay? So I'm thinking in this one, you may not use the malic for the phosphorus, okay? Let's see. Yeah, here's where we use the Haney test. And let me get with you. I'm not sure if they're using the malic also for that. Because if you're going to pay $55, I think you may get that.
So it's still kind of, when you get that health score, it seems,
No.
you want us to write down, when we're taking this test and think about it, but, you could be coming out of corn stalks, you're coming out of beans, you could be doing it now, or you could be doing it May 1st and your cover crop is 6-7 inches tall. It just seems like all that would have a lot of effect on it.
Yes, it changes. It should change. If the test is any good, it better change. It better change. But one thing you're going to definitely see the differences, guys, is this. Look at the difference in carbon in the system. 174, 492. That's a huge difference in the food. There's things I key on, that's huge. That drives the system. So you're going to be focusing on those kind of numbers. That's what you're going to be I don't just only look at this, I look at all the numbers. That's important to me right there. And the ratios are very critical to me. So I look at all of it to make my assessment.
I
But
think the other thing you gotta kind of look at too is that producer that you're dealing with too. Is he willing to go back and put some additional land on later in the season, et cetera? And at that point, then you could actually then delay a test later, at that point.
Yeah.
Versus, if that producer's going to end up putting most of his inputs on early and you wanna get a handle on it, you're gonna have to pull that test before that.
Yeah. Yeah. And see, here's what we're saying, is that we want to provide ourselves another tool so that when we're out there, we can help the landowner better than what we've been using with our university soil sample. It's just another tool in that toolbox. What we've seen so far, and reading a bunch of analysis all over the country, I feel a lot more comfortable about this tool than we've had in a long time. It's encouraging to me. I feel we're a little closer. Now, Rick will tell you, in 10 years, somebody will come and say, we come up with a better test. That's what we're shooting for. We're always striving for a better test. But at least we got something to work with. Let me show you in practical how this has worked. You guys have heard about the Minokan farm. They got a field-sized farm, and I mean, the fields, there's 10 of them. This was all monoculture wheat fallow farm. This is Jay's humor. You know how they found the Minokan, they found the Balkan oil. Now here's the thing when we use the Haney test. Now, we tried the Haney test, and Jay got to meet Rick and all of us, we got it together and says, let's try running some soil samples. Because again, we were using regular university soil samples, and when Jay saw these soil samples and using the Rick Haney test, he was much more comfortable with what he was seeing and what he was observing. I want you to notice the rotations on field 1, 2, and they put a check here, regular treated wheat, continuous wheat fertilized. I want you to keep that in mind. Look at number 3, 4, 5, 6, 10 is the Three Sisters, triticale. These were planted in these fields on field size level scale, okay? From 2009 to 2013. Let's look at how it's showing up. The carbon and nitrogen balance. We want release, nutrients will be released in this band of carbon nitrogen ratio from 8:1 to 15:1. We want to stay within those parameters. Anything over 15, including close to 20, you're going to have nutrient tie-up. It's not, the microbes are not going to mineralize. The more you get it closer to 20, you're going to have some of that tie-up. Remember that field 1? Remember the fertilized one? It ranked number 10 in the organic carbon nitrogen ratio. Look at organic carbon. Continuous wheat, fertilized, ranked number 9. Look at number 2, number 1 in carbon was pea, cover crop, corn, wheat, lentils, corn. Again, carbon. Now let's look at the nitrogen. Look where the nitrogen, oh, that's the phosphorus, the phosphate. The fertilized one was number 4. Total P, inorganic P, organic P. Which was the number 1? The Three Sisters. Interesting.
Do they know what the Three Sisters are?
How many know what the Three Sisters are? Tell them.
Corn, cotton, beans, and some type of squash.
Who's been using the... Yay! We should give you a 5-minute break in a little while. How's that? We're almost done. Isn't that amazing? It's interesting. And then fertilizer has been added to this. Nitrogen. How did it rank? Number 9. Look what the Three Sisters did. Look at the number 1. Let's go back to number 1, number 4. Peas, corn. What's the CC? Cover crop, PP, cover crops. Thank you. Corn, cover crop, corn. That's nitrogen. Here we go. Number 1. Look at the Three Sisters. Interesting, the one that's fertilized, number 9. Now, I want us to think about this. I want us to walk away with this. Folks, last night I talked about this. We have a human desire that we want to predict outcomes. It's impossible to predict outcomes, but we can predict if a system is fragile, robust, or anti-fragile. Natural ecosystems are anti-fragile. Our farming systems are very fragile. Gabe and Ray Steyer's are in a system that are very robust. The journey towards that is called resilience. We want to build these kind of robust systems. What Rick, the Haney test analysis tells us where we're at, but it will not tell you the exact outcome. Let me explain that. Say we took the Haney test, everything looked great, but what if it got dry the last minute? What if you put the wrong, what if you have a heavy, heavy rain and you flushed all the nitrogen out of that system? You cannot predict the outcome. You can predict the exposure. That's the best we can do in science. I think we would be very grown up and be very humble to ever realize that. Once you start studying quantum physics and start getting into the deepness of this, we'll never know all the mysteries of it. But if I can know where I'm at, then I can feel comfortable in management. That's what we're shooting for. Again, we're done. Let's wrap that up. Again, okay, we'll talk about that later. Guys, any questions so far? And we'll take a break. Let's take a 10-minute break. Is that okay with you, Rick?
Yeah.
Let's take a break. Any questions, guys, before we take? Because you guys were brave right after eating and had to sit there and listen to me. And let's go ahead. Go ahead.
When would you like to take the Haney test? If you know, would you rather do it in the fall, rather do it in the spring, late spring? I mean, what's your ideal?
My thing for me, guys, if the biggest the thing for me is like if I have a farmer that wants to do a benchmark, I would take the most activity happening in the system is in the spring, okay? In the spring. But for me, is if I'm going to go out there with a guy that's fertilizing corn, he's going to do a couple hundred acres of corn, my goodness, I'm going to go take that bit right before V10 stage. I'm going to go 2 or 3 weeks prior to that because I've got to give myself some leeway. Because I know at the V10 to the R1 stage, you're going to demand huge amounts of nitrogen. Right, Jeff? That's the biggest load for nitrogen. So I want to find out, because the biggest expenses these guys have is the nitrogen costs. So I would want to do that at that time. So I would back it off, give me 2 or 3 week window. Before we reach the V8, V10 stage, I'm going to go out there and get a sample. I want to know how much nitrogen the system's going to have at that point in time. It's a rule of thumb. And then that's what I would do. And what else would you add to that, Jeff? Anything?
Well, as I was talking about before, if you're working with somebody and he says, well, I'm not going to mess around with side dressing in at that point.
Yeah, if he doesn't want to side dress either. That's for side. Thank you, yeah.
And he's going to put it on earlier, I mean, that's when you go out.
Yeah. If you're going to put it out there and you're going to put it out there earlier, I like side dressing, but some people won't do side dressing.
Most of our guys would be more apt to put on a principal end and then come back with side dress.
When would they do the principal in?
They're going to do that in the fall or early spring.
Are they going to use anhydrous?
Yep. And I know when I've talked to a lot of guys that are into cover crops, they seem to move away from anhydrous and go more with so much at planting time and then side dress.
But if they take the soil test at the time when it's most needed,
Yeah.
maybe it's not going to impact the application this year, but if they take the soil test when it's most needed at your V10 and it shows gobs and gobs of excess, then what should you do for your fall anhydrous? Cut it down some.
I think one of the things we're definitely seeing throughout the whole country is way over fertilization. Way too much. Especially on our no-till guys. But the no-till is getting healthier and healthier. We just put way too much. And then dairies, when we're using no-till and cover crops, oh my goodness. I have farmers that are still in the Chesapeake that are still fertilizing with manure. And there's no need. And they're doing multi-species cover crops and their soils look fantastic. We're still doing that. So, but you got to, we also got, and I'll say this last statement, guys, farmers are risk averse. They're not going to take a risk on yield, and they figure, I'll spend a little more on fertilizer. And they will do that every time. And I'm saying, no, you're going broke, and it doesn't really help the soil ecosystem either. Too much nitrogen, too much phosphorus. If I have farmers already pushing close to 200 pounds of P, I said, stop putting manure on. Stop putting the manure on. You're shutting down mycorrhizae. The literature shows anything 150 to 200, you're going to start hurting sporulation and mycorrhizal buildup. You're going to hurt the system. And now you're going to have a problem with phosphorus being taken up. So that's where we're at. Go take a 10-minute break, guys. You guys need it. I can see it in your face.